Good morning everyone. So after this talk on mitochondriopathy, the second topic today deals with more of epidemiological issues, where we are going to discuss about the recent outbreak of severe acute hepatitis in children. Is it a new disease or is it just old wine in a new bottle? So in April 2022, for the first time, WHO reported a sudden surge of severe acute hepatitis cases from several countries in Europe and from USA. And these children were usually younger children with high AST/ALT, a prodrome which was mainly in the form of GI symptoms, and the normal hepatitis A to E viruses were negative in these children. Now, these cases were labeled as severe acute hepatitis, and a working definition was given by WHO, where the transaminases had to be more than 500, and we had to exclude all other causes of known acute hepatitis. And if INR was more than 2, these cases were labeled as severe acute hepatitis with ALF, but majority of these reported cases were severe acute hepatitis without ALF. After this reporting by WHO, there was flurry of cases. Everyone started reporting these cases, mostly again from Europe and USA, and hardly any cases, very few cases from Eastern Asia. So by now, more than 1,500 cases have been reported from more than 35 countries over last 1 year, and several incriminating pathogens have been described, including adenovirus, AAV2, SARS-CoV-2, human herpes virus 6, Bartonella, or something that it is some novel pathogen and a novel variant of a existing pathogen which is causing this epidemic of a sudden surge of cases. So 3 questions arise in my mind which we should be able to answer by the end of this is, was there an actual surge in the cases of severe acute hepatitis compared to previous years, or was this just an effect of lockdown being lifted after the COVID pandemic? 2, if yes, what was the cause? And 3, what is the outcomes? So coming to the first question, was there a surge in cases of severe acute hepatitis compared to previous years? The data from the US show that in the corresponding, in 2021 to 22, there was a 2-fold surge in cases compared to the same period in the last 4 to 5 years. And in this surge of cases, it was mainly adenovirus and SARS-CoV-2 that was being detected with greater frequency than what it was being detected in the previous 5 years. However, the data is not consistent, and this Eurosurveillance data shows that only 5 countries listed here showed a increased incidence of severe acute hepatitis as compared to previous 5 years among the 17 surveyed European centers, and this was even lesser in non-European centers, where only 1 center reported an increased incidence of severe acute hepatitis. What about in India? We tried to search our cases, and we have published this recently. There was an increase in acute hepatitis. Now, we are not labeling this severe acute hepatitis. This is acute hepatitis. 177 cases. This is higher than numbers reported between 2020 and 2021 that is expected because of the effect of lockdown, but it's not higher as compared to 2017. We do the extensive etiological evaluation only in the cases of acute liver failure, so I am taking only those cases of among these acute hepatitis who are presented with acute liver failure, and we find that those with unknown etiology, the number has remained more or less constant if we compare from 2017-18 to 2022, and the outcome has remained same. So, to answer the first part of question, yes, there was a surge, but it was there only in some parts of Europe and US, and definitely not in India, because by now many more case series or case reports must have come up if that was the case. Now, if there was a surge which was there in some parts of Europe and US, what was the cause of this surge? So this is an important study which came in recently, that the first chart shows the cases of severe acute hepatitis over time. The second and third chart shows the virus detected in the wastewater in Dublin, Ireland over time, and we can find that a rise in the red and the pink and orange are the AAV2 proteins, and the black one is adenovirus. So you can find that there was a rise in detection of AAV2 and adenovirus F41 in the wastewater, corresponding to the same time period when there was an increase in severe acute hepatitis cases in United Kingdom. And this peak did not correlate with other viruses like SARS-CoV-2. So this made them think that these two could be the possible pathogen responsible for severe acute hepatitis in the UK. This again investigated the various viruses. Several viruses were investigated in the UK pandemic, and you can find green is the positive, red is the total tested, and you can find that more than 50% of cases, these cases tested had adenovirus in the blood or plasma, and only about 20% had EBV and 10% had SARS-CoV-2. The other viruses were not significantly increased. And important thing was, among the adenovirus F41 was present in 92%. Now, if we compare this with the natural history of adenovirus that we know of, we know that adenovirus usually causes a self-limited upper respiratory or GI infection in immunocompetent children, and by the age of 5 to 10 years, most of the children achieve serotype specific immunity for lifetime. Cases of acute hepatitis have only been reported in immunodeficient children or immunodeficient adults, mostly those post-transplantation. Whenever it causes acute hepatitis in immunocompromised, the liver histopathology shows definite necrosis and intranuclear viral inclusions, and the adenovirus is always detected in the liver. So in contrast, if you compare this current outbreak of human adenovirus with what has been reported and known from the literature, this surge or this outbreak is happening in immunocompetent children. Human adenovirus has only been detected in the plasma and not in the liver histology specimen this time, as compared to what has been classically described. The outcome is good, whereas the outcome with acute hepatitis of adenovirus is known to cause severe hepatic necrosis with fatal outcome. And this time it is adenovirus F41, whereas the previous reported predominant serotype is 5. So all this makes us think that yes, adenovirus is being detected in the blood, but this is not possibly the cause of this outbreak, or at least not the single most prominent cause of this outbreak. Now coming to the second virus that has been incremented is AAV2. Now AAV2 we all know has been used as vector for gene therapies, and we know when administered at very high doses, it has led to fatal outcomes with these gene therapies. And liver injury with AAV2 is immunologically driven both by T cell and innate immune mediated response. Just for this, AAV2 are actually parvovirus, which are only single stranded. So they are actually defective virus which require the presence of a helper virus, which could be adenovirus, HHV6 or EBV, to cause an injury to the humans. And AAV2 is the commonest subtype, and this is by genomic studies we can identify the subtype, and AAV2 is the commonest subtype of AAV. Now, two studies came in Nature in March 2023, two studies in the same issue of Nature, and both dealing with the same, this first study is from the United Kingdom and the second is from US. Here they have taken 38 cases of severe acute hepatitis and matched them and compared them with the age-matched controls by PCR, metagenomic reads and viral whole genome sequencing coverage, and they found that AAV2 was significantly higher in these cases of severe acute hepatitis, both in the liver tissue as well as in the blood, as compared to adenovirus and HHV6B, which were found in very low concentrations. And AAV2 DNA was found in close to 100% of these cases. And the phylogenetic studies excluded the emergence of novel strains of these viruses. Now the second study again came in the same issue and in the just next subsequent. This took 16 cases of severe acute hepatitis who had already detected adenovirus in their plasma and compared these with controls. Now important control is this first one, where the control group is acute gastroenteritis caused by adenovirus. So they are comparing acute hepatitis caused by adenovirus with acute gastroenteritis caused by adenovirus, and they find that almost 100% of the cases had AAV2, whereas none of the controls, that is adenovirus related acute gastroenteritis, none of them had AAV2. Similarly, close to 85% had EBV also present in them, whereas none of the controls had this. So does that mean that, and the third part is the serotype. The adenovirus type 41 is more common in acute hepatitis, whereas other serotypes were there in GE cases. So that possibly means that AAV2 in combination with adenovirus F41 or in combination with another helper virus which could be EBV or HHV6 causes acute hepatitis, whereas just adenovirus without AAV2 causes acute gastroenteritis, self-limited acute gastroenteritis. This is the same thing. The third virus that has been incriminated is SARS-CoV-2. There are various reasons to believe that SARS-CoV-2, there's not reasons, but there are several mechanisms by which SARS-CoV-2 can cause acute hepatitis. We include direct cytotoxicity, a cytokine storm, hypoxic liver damage, vascular damage by endothelial inflammation, bile duct injury, modification of gut microbiota, and drug-induced liver injury by drugs given for this COVID pandemic. But there are reasons that to not believe SARS-CoV-2 as the cause of this outbreak is that the peak of acute hepatitis has occurred beyond the peak of pandemic. Yes, you can argue that it coincides with the Omicron variant, but Omicron variant has persisted for a longer time, whereas this peak has gone and had come and gone away. There is no evidence of direct liver infection shown in the form of from the liver histology, at least with the Omicron variant. Deranged LFT is most commonly documented in adults with SARS-CoV-2 and less commonly in children. And this outbreak was mainly described in young children and especially less than 5 years old. So again, that is against this. And SARS-CoV-2 positivity rates, this is the most important, SARS-CoV-2 positivity rates in children with severe acute hepatitis was not higher than SARS-CoV-2 seropositivity rates in the general population. Whether the vaccines were responsible, there is this hepatology paper, multicentric, which shows that 87 patients with vaccine-induced hepatocellular injury caused by all types of COVID vaccines, and 57% of these patients were having features of immune-mediated hepatitis and they responded to steroids. But only one of these had ALS. But thing with COVID vaccine is most of the children who were younger children had not received COVID vaccine by the time they developed severe acute hepatitis. So this theory that COVID vaccine was causing this outbreak also looks unlikely. And from the US data, only 4%, and from the European data, 19% of these children had received COVID-19 vaccine. Apart from these three viruses which have been most commonly implicated, there are other viruses. I am not going into them. But there is definite evidence of immune etiology whenever the etiology is not known in the form of increased dense CD8 and CD103 infiltrates in these cases as compared to the cases with a diagnosed cause of acute liver failure, suggesting that there is an immune-mediated injury that is going on. And the recent paper shows that the children who developed severe acute hepatitis had HLA DR4, suggesting that there could be a susceptible genetic background on which this adenovirus and AAV2 could be leading to this severe acute hepatitis. So what is the possible, from all this, what is the possible cause of this outbreak of severe acute hepatitis seen? So from the data that I have shown, it is likely that AAV2 in the presence of a helper virus, which is either human adenovirus type F41 or HHV6 or EBV, in certain genetic susceptible individuals is leading to an immunological T cell and innate immunity mediated injury leading to acute severe hepatitis. There could be a role of a super antigen like from the COVID antigen, but that is not proven as yet. What about our Indian cases that have been reported? Now, I am presenting the data only from the cases of acute liver failure, those cases of severe acute hepatitis with undefined etiology who presented an acute liver failure, because as I said, just in acute hepatitis, we have not done all the virus panels to say that there was an undefined etiology. So among these cases, we can see that most of our children were very young, between 1.5 to 4 years of age. They had a predominance of GI symptoms. You can see this was different from what we were finding previously, and this made us think that possibly we are dealing with adenovirus, which causes a predominance of GI symptoms in very young children. So we went ahead and tested adenovirus in blood and stool of all these patients, but we couldn't find adenovirus by PCR in any of the 8 cases of acute liver failure with undefined etiology. We also tested SARS-CoV-2 antibody, as we are getting reports that this could be the possible reason. And surprisingly, we found that uh, five out of 7 had a SARS-CoV-2 antibody, that is close to 75%, and these were not vaccinated children, these five. But when we compared it with the seroprevalence data of SARS-CoV-2 antibody in the Indian population, which is published in the general population, we find that this is not significantly higher. Features of autoimmune hepatitis again were not there in most of these cases. I would like to point about this case 1, 14-year-old child who presented to us and had a SARS-CoV-2 antibody sample cutoff ratio of 5.28. This child has received COVID vaccination 15 days prior to onset of jaundice. All other causes of acute hepatitis were ruled out. She had elevated IgG and ANA positive. Liver biopsy showed features of autoimmune hepatitis. She was treated with steroids, but she developed neurological worsening all of a sudden, and then underwent liver transplantation. And post liver transplant, she had a poor neurological outcome with diffuse findings in the MRI brain, which was again not explainable. Whether this was a cause of the vaccine-induced immune-mediated damage is open to question, but this case matches with the cases described in the hepatology paper previously. Coming to the last part, that is the outcome. What is the outcome of these patients? Now, whenever severe acute hepatitis is without liver failure, is with good outcome with close to 90 to 95% native liver survival. But when presenting with liver failure, it is associated with poor outcome. There is only one paper. There are only two papers. One is this King's College paper where they describe 25% native liver survival, and in our paper also we showed that there was only 25% native liver survival, and only those children who never progressed to grade 3 or 4 HE survived, whereas all those who progressed to grade 3 to 4 HE either died or underwent liver transplantation. Another important feature in these children was a very fulminant, refractory raised ICP. All of them had increased ONSD more than 4.6 mm, required mechanical ventilation, and all of them required thiopentone beyond propofol, bridging therapies. And important thing is, the 4 children who died within 1 to 2 days of admission. That means they don't give enough time even for you to evaluate and prepare for a LT. 2 children underwent liver transplantation at 8 and 14 days respectively. Histopathology, we had 6 patients. There was just massive hepatic necrosis in all of them. So comparing this group of indeterminate ALF or severe acute hepatitis with ALF with what we commonly see is hepatitis A induced ALF, we find that these children were younger, had a predominance of GI symptoms, had more severe disease in form of higher INR and poor native liver survival and a short time to death. Important one thing that I would like to point out, we are also getting an increased number of acute hepatitis cases in 2022, which is comparable only with 2017, and the cause of surge in our cases possibly is a surge in the number of hepatitis A, as we can see that the IgM-HAV positivity rate among acute hepatitis had suddenly shot up in 2022. So I would like to at least believe that the surge in cases of acute hepatitis that we saw at ILBS in last year 2022 was due to a surge in hepatitis A and not due to adenovirus or AAV2. Following these reports, we have now modified our diagnostic protocol since May 2022, and in the cases where we don't get the first line markers, we are now testing all the possible viruses like parvovirus, adenovirus, SARS-CoV-2, HHV6, EBV, CMV, etc. But we are yet to find these viruses in the samples. So it is possibly only and only HAV that is causing all the surge in India, in North India at least. So to conclude, there is a surge of acute hepatitis cases in 2022. This surge in US and UK is multifactorial and is because of AAV2 in association with a helper virus like adenovirus, HHV6 or EBV, and is an immune mediated process in possibly genetic susceptible individuals. In India, this surge is most likely due to hepatitis A virus, as the children were suddenly exposed to this virus after opening of the lockdown. And the children with severe acute hepatitis of unknown etiology who are presenting to us as ALF are younger children with hyperacute presentation, predominance of GI symptoms, and in this scenario, they have a fulminant course, and hence we need to prepare for LT in a very expedited way. We have looked for adenovirus and HHV, but we also possibly need to look for AAV2, which we have not looked for in our cases. Thank you.